Technology principles and processes. So competent host for transformation with recombinant DNA. So competent host, the host need to be competent so that the transformation can be done, and we want to transfer the recombinant DNA. Okay, so this is the competent host for transformation with recombinant DNA. Now two types, like microinjection in animal cell, now biolistic and gene gun in plants. So micro injection in animals and biolistic or gene gun in plants. Now process of recombinant DNA technology. So first step, that is the isolation of the genetic material. Genetic material, it is DNA. So we use the lysozyme for bacteria, cellulose for plant cells, chitinase fungus for fungus. Now DNA that separates out can be removed by spoiling. So spoiling, it is a process where DNA can be separated out from these other biomolecules. So this is the isolation of the genetic material. This is the first step. Now in the second step, the cutting of DNA at specific locations, we use the restriction enzyme for this. Now third step, that is the amplification of gene of interest using PCR. So the gene that we want, that is the gene of interest, so we need to amplify it using the PCR. Now PCR, what does it mean? That is the polymerase chain reaction. Now three steps in the polymerase chain reaction. You see, each cycle has three steps. So each cycle, first one is the denaturation, second is the primer annealing, third one is the extension of that primer. So annealing of the primer, you see, like it is 5 days to 3 days, it is attached on both the stands. So denaturation heat, we separate both the strands. Now using the DNA polymerase, this is the enzyme, that is tag polymerase, and this enzyme works at the higher temperature, plus deoxyribonucleotide, so that the change gets elongated, right? Now this is the extension. Now all these two things use in the extension, so this can extend this strand. So the after the 30 cycles, amplified 1 billion times. So after 30 cycles, the resultant is the 1 billion. This is the amplification. So this is the amplification of gene of interest using PCR, three step, and denaturation, annealing, and extinction. That's it. Now the insertion of recombinant DNA into the host cell organism. So insertion of the recombinant DNA into the host cell organism. So the ampicillin resistant gene in this case is called a selectable marker. So insertion of recombinant DNA into the host cell using the selectable marker. Right now, obtaining the foreign gene product